So, hi everyone, my name is Lotte, and as you just heard, I'm not only a psychologist, but I'm also a biomedical researcher. And today I would like to address a topic that is often considered a psychological problem, whereas it also has a major biomedical ground, and that is stress. So, I would like to start off by asking all of you a question. Who of you has ever experienced stress? See, that's kind of what I expected. But now I would like to ask you a little more personal question. Who of you has ever felt sincerely ill by this stress? See, that's already a lot less. And that is exactly the concept of stress vulnerability that I would like to talk to you about today. So we already know a lot about the environmental factors affecting mental health, but we do not know so much, unfortunately, about the molecular basis of this and what is actually affecting our stress vulnerability. So what happens when we're exposed to stress? So imagine you're exposed to a stressor. Let's say you see a snake in the backyard. I mean, I know this is not very likely to happen in Germany, but whatever, imagine you see a snake in the backyard. The brain somehow registers this, and by sending out a lot of molecules, it lets the adrenals eventually secrete the stress hormone cortisol. And the stress hormone cortisol, it puts our body in some sort of state of alertness, and it tells us to either run away from a stressor or to fight back. But as many processes in our brain, this needs to be regulated, or in our body actually, this needs to be carefully regulated. Because if it does not, we are always going to be in some sort of state of mind of alertness. And we don't really want this to happen when we figure out that the snake was just a stuffed animal of our little nephew. So you might think, there are so many different factors involved in this process, in so many different parts of this cycle. So where do we begin? Well, I give you an answer. We begin at the FKBP5 gene. And I will tell you tonight why this is our favorite gene and why I study this in the vulnerability to stress. So what do we already know about FKBP5? We know that it interacts with childhood adversity, because from human studies, when they tested a lot of different people who had similar type of childhood adversities, they actually saw that only a very particular group of people that had a specific variant of this FKBP5 gene that led to higher protein levels of FKBP5 had a much higher risk of developing major depressive disorder. So this is quite fascinating. But what is actually going on inside our brains then? What is the molecular basis behind this, and why is this happening? So we want to figure this out, obviously, by studying the brain. And we have a lot of cool tools these days to study the human brain. However, these are mostly correlational. And if we want to know the cause, we would have to randomly assign a human person to a stress condition, and that would be quite unethical. Let alone if it would change people's genes. So that is why I work with animal models. And you might wonder, what do you do with these animals? Well, we genetically manipulate them. So what we can do is we can either cut out this FKBP5 gene from their DNA, or we can overexpress this gene, which creates more protein. And then what we do is we can study the brains and their behavior, which might relate to human behavior. So what we do is we expose these mice to stress, and then we expose them to, for example, an anxiety-like behavior test. So this is the most common test that we use. It's the elevated plasmase. And as you see here, oh, sorry, I'm standing in front of it. This is the closed arms. The blue area is the closed arms, and the yellow area is the open arms, so to say. And, oh, just going back. So if I start this little video of this cute little mouse, I can explain to you what is going on. So when they are inside the blue area, there's the walls, it's dark, it's kind of nice and safe for them. But if they're in the open arms, I mean, they're quite curious, so they want to go out, but it's very scary for them. So what we can compare is we can compare the animals that like to stay inside this blue area, the safe area, or if they go out into this yellow area. And as you can see, this animal is quite a daredevil. So what do we study? Well, our lab has already done a lot of experiments on this, and what they found is that when we overexpress this FKBP5 in a brain region that is related to fear and anxiety, the amygdala, we saw that there is an increased anxiety because they spent less time in the open arms, as you can see here. But when we block this FKBP5, there's actually more time in the open arms, which means that their anxiety behavior is reduced. And when we cut out the FKBP5 gene and we expose the animals to a stressor, we actually see that they're less vulnerable to this stress than in wild-type animals. But what is now eventually the ultimate goal? Well, the ultimate goal is, at first, to figure out what's actually happening in the brains on a molecular and on a structural level. But eventually we want to, of course, translate this to the human brain, the patient brain, what it's all about. And for me, as a translational researcher, knowing both sides of the case, I think it's so important that we combine this molecular research together with the research in the clinic. So, one last message. I hope that I convinced you tonight of how important it is to not only study the environmental factors, but also to study the molecular basis. And I'd like to give you one last message to take home. Just keep calm and let us study the molecular basis of stress. Thank you.